This is Israeli Technology Founders Speak, a podcast of conversations with successful Israeli high-tech and biotech entrepreneurs. With your host, Avraham Hermon. Steve Grun is the co-founder and CEO of MNDL, an Israeli startup that uses AI to design better DNA sequences for making proteins, helping companies produce up to 20 times more protein than traditional methods. Their technology takes the guesswork out of gene design for biotech and pharma companies, developing new drugs, enzymes, and other biological products. Avraham sat down with Steve to discuss why most biotech companies struggle to produce proteins at scale and how Mendel's AI platform fixes that. How he turned 15 years of academic research into a SaaS product, their $2 million pre-seed investment, tips for startup founders, where he sees his company in 5 years, and much more. This podcast is a creation of J.M.B. Davis Ben David, an intellectual property law firm serving clients around the world. You have great innovations. We keep them safe. It's not enough to just have a great startup idea or innovation. If you don't legally protect your innovations, products, and brand, anyone can claim them as their own. We keep your great innovation secure. Learn more by going to jmbdavis.com. That's jmbdavis.com.
I'm sitting here with Steve Grun from MNDL, and we're in their Herzliya offices, and we're talking a little bit about their technology. So first of all, we're going to talk about recombinant proteins. So what are recombinant proteins and why are they important? Just for our listeners, just to give some background.
Recombinant proteins are a cornerstone of modern biotechnology, and to understand what they are, we first need to explain how cells work. Every cell follows a similar process called gene expression, which you can think of as the cell's manufacturing pipeline. So there's DNA, RNA, and proteins. DNA is the long-term blueprint, RNA is the temporary copy of one instruction, and proteins are the final products. And they do almost everything in the cell. So this flow, DNA, RNA, protein, is universal to life. Now, biotechnology allows us to take a DNA recipe for a useful protein, let's say insulin. Insulin, for years, was produced in pancreas of porks and cows, and they were killing a lot of porks and cows in order to take the insulin. Now, a couple of decades ago, it was synthesized. And why is it synthesized? So they took the real insulin target gene or the protein and they inserted it into another organism. So let's say insulin, an antibody or an enzyme, you can place it in another cell, often mammalian cells, yeast, bacteria, even algae today. And that cell becomes a tiny biological factory that makes the protein for us. So it's a real factory.
So you mentioned insulin as one of the proteins that can be made with this technology, but from what I understand and being in the field, a lot of the drugs that are out on the market today, a lot of them that we know as biologics, are made through this process, right?
Yes. Major part of biologic, as you said, like antibodies, antigens, enzymes, hormones, and diagnostic proteins are produced through this process of recombinant protein. So it's a market of $850 billion today.
Impressive. So that's basically a lot of the new drugs that are coming out on the market are made through this technology, whether they're cancer drugs or drugs for autoimmune diseases, like a whole variety of diseases are treated today with biological drugs, right?
Yes, totally. Cancer therapy, vaccines, even food tech, enzymes in food tech. Even when you're washing your clothes, you're using enzymes. And those are produced through recombinant protein production.
Interesting. So let's talk about what Mendel solves. What's the problem that you guys address?
So when you're combining a foreign gene, a target gene, and you put it in a host organism like yeast, insect cells, mammalian cells, and other host organisms, they are not speaking the same language. So what's happening is that the yield, the production yield is very low. And that's why 80% of recombinant proteins fail to scale. And that's where we come in. So we are using an AI-driven platform, is optimizing the gene sequence of the target gene and tailoring it to the host organism. So it's different gene sequence if it will be on yeast, if it will be produced by an E. coli, another bacterium. So we are tailoring it to the host organisms. And then we can get to yields of 10 times the quantity that was before, even 50 times. And that's making a huge difference because this is the difference between success and failure. And that's why without Mendel Bio, 80% of recombinant proteins products or projects fail to scale.
They fail to scale and they just have to try again in different organisms, or they have to just start again from zero?
It's a good question. There are 50% of the expression already fails in the beginning. So then you get into a process of DBTL. It's design, build, test, learn. And this is like a lot of iteration that is costing a lot of money. So sometimes we speak about startups who are producing recombinant proteins. The money is finished. So they got to a certain point and there's no money left. So they're closing the company. Or you've got a budget of a biologic that is developed, a recombinant protein in an established company. And unfortunately, they didn't get to the economical quantity that is needed for this product. So having a product that can be profitable. And then, then the project is closed and the product is going to the drain. So this money just was wasted.
Right. Okay. So what you're going to do is you're going to cut out all that wasted effort and time by using your AI model?
Yeah. So we're making biology more predictable. So we will give the customer their protein. So we are not developing their protein sequence. They're giving us the protein sequence that works for them, but it doesn't give enough expression in the host organism. We will use our models, our AI, and all the models that were developed and algorithms, and we will give them a couple of variants of different gene sequences that they can try out in their lab in order to get to better expression. So we are narrowing the search space. Instead of having like 10 to the power of thousands possibilities that are sometimes tried out through high screening throughput and using like really millions of dollars to try to check all the possibilities, we are narrowing the space to 10, 15 variants, maybe a couple of iterations, but it's cutting time, cost, and at the end, you've got a profitable product that's getting much faster time to the market.
Okay, that sounds good. Tell me how the idea was born. Who came up with the idea?
So our CSO is Professor Tamir Tula from University of Tel Aviv, and he has researched gene expression for the past 15 years. And his lab with him are studying every phase of the gene expression. So as we said in the past, we said DNA, RNA, and protein. It's very simplistic. There's a lot of phases inside. Transcription, translation, folding. And then there's problems of genetic stability. The idea started with the research of Professor Tamir Tula. Our CSO is from the University of Tel Aviv. He has been researching gene expression for the past 15 years. He's leading one of the largest synthetic biology lab in the world. And saying that gene expression is DNA, RNA in protein is very simplistic. There's a lot of phases inside. Transcriptions, translation, secretion, and then there's the results of problems of genetic stability, of co-translation folding, how the protein folds. And every phase can be a bottleneck. So when you are building algorithms and taking into account all problems that can occur, then you can get to a product or a service that can give a gene sequence that it's much more efficient and scalable to the customer. So I was in an incubator, the kitchen from Strauss in the past, and they had a couple of companies using precision fermentation and cultured meat. And I could see the issues that those companies were confronting. Very great ideas, but at the end, they had a lot of problems to scale. And when I was looking for my next idea in a startup, we were looking at, with my partner, Avraham Miller, we're looking at what technology can help this. And we got to Professor Tamir Tula, and we understood that he developed a state-of-the-art technology in order to bypass those problems and get to predictable, scalable gene sequences. And what we said, instead of him doing this work for months for customers, we were able, and that was the idea, to include everything into a SaaS online platform where every scientist in the world can put his gene sequence, choose his host organisms, and just with the pushing of a button can get a couple of variants that can change his life, change the life of his project, and get to a scalable gene sequence.
How did you speed that up so quickly to the press of a button?
It's 15 years of research that were built on an academic scale. So what we did, we translated it in a totally robust system. We translated the algorithms, we looked at all the bugs and everything that's possible. And at the end, less than a year, we got to an intelligent layer. We call it the intelligent layer of synthetic biology, where automatically you say, I want 5 variants, 10 variants, 15 variants. And the system with a rule-based algorithm is choosing which one or which gene sequence is tailored to the customer and tailored to the host organism of his choice.
So what stage are you in now? Is this service available for startups and for companies to use?
So the online SaaS tool is available on a beta version today and available for every academic scientist in the world on RUO base. So you can use it, get into the app, put your gene sequence and host organism, and you can get those variants just in a couple of hours or even a couple of minutes and use it afterwards in your lab. And then we are expecting also to get the information back so we can do another iteration. And this data flywheel is helping us also to train our models. So our models getting much better, much more robust. So that's the idea for us of combination of biology, AI, and a SaaS online tool in the middle. This is just together creating a very interesting system of generating more data and the AI extrapolating predictable sequences or better sequences based on all the data sets that we are gathering.
Can you tell us a little bit about how you combine these two very different fields? On the one hand, very high level biology, synthetic biology, and on the other hand, AI?
Yes, for sure. So one is helping the other. So based on our models that have been developed in the university and translated into the company robust system, we are able to generate gene sequences that are qualitative. So it's not that we are giving the customer something that he will fail with. We are increasing the rate of success. So when he's getting the gene sequence, there's a high ratio of success. So that's good data points that we can get back. After, when we getting this data flywheel together and we bring those information back, then we can train the system, as I said before. And then the AI can extrapolate more and more data points or all these models that are making the tool even much more predictable.
And how do you combine the two disciplines that are very different from each other? In other words, you have people that come with a background of AI and you have people that come with a background of biology. How do you talk to each other?
So first, we are employing bioinformatics. So people that have got a background also from data science and biology.
Okay.
So this is already combining. We are employing people that have got a biology background. So bringing somebody with only AI will be taking time to learn what the company needs. So we are always happy to bring people with experience that already have developed new novel LLM models, other vertical AI systems. So we are looking at ourselves as a vertical AI for biologics in a certain way. So combining all those forces of data science and biology and AI is giving us an unfair advantage when we're looking at the future.
Right. Okay, let's talk a little bit about your investment. This obviously, your system sounds like it's complex and it took a lot of capital to get it started, get it running. Can you talk about that a little bit?
Yes, for sure. First thing, my background coming from companies who, let's say my last company was a company in the food business a couple of years ago. And I managed to work with small budgets. And that's something that is important for my background. So when you're managing a startup, you need to always look at the cash flow. So we're managing to really do a lot of things with little budget. So now we raised $2 million as a pre-seed from Earth and Beyond Ventures, a leading deep tech investor in Israel, with the support from the Israeli Innovation Authority. So they are managing an incubator here with companies from quantum computers and space technology, so very deep tech sectors. And with this investment, we recruited a great team, really specific people, as I said, with background of data science and biology. And we are able, in less than a year's time, to translate this technology academy into a robust system and already put it on the market as a beta version. Now we are looking at getting to the next stage. We'll be raising a seed round in the next couple of months in order to streamline our development, increase the users of our SaaS online tool, to integrate it in the workflows of biopharma companies. And what's really important for us, and we're doing it every time, is that every case study that we are working on is giving us support to present it to another company. So
I see.
When you have got a company that is, let's say, producing antibodies in E. coli, they don't want to get a case study about antibodies in CHO. They just want specific mammalian cells. They're looking at something that really specific experience to what they do. When you've got a big company, no one will burn his career pushing us. They need just to be convinced that we can bring value to the R&D, and then they will really push it further.
So the more relevant case studies that you have, the easier it will be to market your product.
Totally. So that's what we're working on, increasing our portfolio of work in order to open it to customers and take down the risk aversion, because biotech companies are risk averse. Okay, don't want to start with somebody that they don't know. So we need to lower this risk feeling for them.
I see. So it seems that you are, on the one hand, using your customers' IP and their ideas of recombinant technology, but then at the same time generating new intellectual property relating to the variants that you suggest to them. How does patents and other forms of intellectual property work with your company?
So we've got, first thing, we licensed the technology from the University of Tel Aviv.
Right.
So we've got 5 patents families that are helping us, one, to protect our technology, but there's a lot of trade secrets also. So a secret sauce, let's say. So it's not because of the patents, you really read in the patents that you now can solve all the problems that we are solving. It's really a mixture of IP and knowledge.
I see.
And that's our success factor, bringing people with experience, but having this IP that is protecting us. And we are generating more patents, more work on other host organisms. So we are expanding our portfolio and expanding our potential of increasing our sales in that way.
I see. So our listeners will know that the previous episode is with the Innovation Authority, and we talked a lot about deep tech and the Israel Innovation Authority's interest in deep tech. But let's talk a little bit about how being in Israel impacts your company.
I'm originally from Belgium, moved to Israel 25 years ago, have been in the industry for 25 years here in Israel. Obviously, it's, as everybody knows, a real entrepreneur-minded country because there's no too much raw materials here. But it's important to say that entrepreneurial mindset is not enough if you don't have the support. And this mixture of getting the Israeli Innovation Authority helping you in pushing your ideas, bringing the cash for the beginning is helping a lot. I think there's a lot of, let's say, a jealousy outside Israel looking at the ecosystem that we've got here, because the Israeli Innovation Authority is giving support that no one gives in the world.
Interesting.
And
so that complements our last month's episode very well, because the Innovation Authority actually spoke about their contributions, and now we're hearing it from the field that this is indeed their contributions are helping and are unique when compared to in different countries.
So there are great innovations here. Academy, Weizmann's Institute, Tel Aviv University, Technion, that's one. There are great people, great minds. And one of the most important things is the resiliency. And I know that Israeli people felt a lot of issues in the last couple of years with the war and so on, but development continued. Companies continued. Despite the war, companies continue to succeed, investments, and we now hope that really now we're starting to have a really blooming era.
Yeah, I hope so. Sounds good. So what are your biggest hurdles moving forward as a company? It sounds like everything's been pretty good so far, right? You got investment, you have their product moving forward. What difficulties have you had moving forward?
So a couple of things that are very specific to us is that gene expression data is very limited and very noisy. It's not AlphaFold when you're looking at the information with AI from all kinds of databases and so on, and we're able to predict the folding of a protein. Here, there's much less data. So, as I explained in the past, when we are creating own database of information through our data flywheel and combine it with the AI models that we are developing, then we are increasing the data that we need in order to give the best services and produce the best libraries of promoter or signal peptides that we want to develop. That's one hurdle, is that biotech companies are very cautious. So we want to show them a portfolio of works in order to decrease their risk feeling. And that's why we are building strong case studies, and that's a main part of our roadmap. And finally, we've got already our model trained on 10 host organisms in our system. We can increase it. That's not a problem, costing time and money. But every organism is different. So expanding our models to support more hosts, like more than yeast, bacteria, plants, will open new markets for us. So that's the next hurdle that we have. And that's why we need also data and experiments.
What tip would you have for a founder that's looking to start a company with a new idea?
First thing is, don't get in love with your technology. Most important is start with a real problem. So if there's no real problem, your technology won't help. That's one.
So you need to have the need for it and the problem that you guys are actually solving.
Secondly is talk with customers early. So you're developing something. Are you sure that's what they're expecting, that's what they need? Instead of burning your cash on developing already a prototype, advanced prototype, speak with your customers, show them an early prototype, and they will give you feedback. So that's important. Next thing is build a team with complementary skills. And next thing is move fast. Move fast, faster, even faster.
Like what you said, you have a limited amount of resources. You need to know how to move quickly and use it efficiently.
Yeah, totally. But always ready to adjust constantly. So there's always feedback. Listen and change when needed. Otherwise, you will be going fast and hit the wall. So it's important to be agile and change your strategy sometimes. Yeah. And most important thing is resiliency. Stay persistent. Deep tech breakthroughs take time, but the impact can be huge.
I see. So patience.
Yeah.
Good. These are a lot of tips, and they sound like very good tips. So hopefully our listeners will benefit from that. Where do you see Mendel in another year or another 5 years down the road?
So in 1 year time, we see Mendel as a widely used SaaS platform for gene design and expression optimization. So that's why we're hiring people just to do that today. User acquisition. Having strong case studies across biopharma and enzyme developers, again, in order to make it easier for us to get more projects, and expanded support for more organisms and production hosts in order to increase our market potential. So that's what we see in next year. And our dream in 5 years' time is being a global leader in AI-driven DNA design or DNA engineering.
But not just specifically for this problem. You're saying other problems as well?
Yes, more problems, also give out other solutions. So not only optimizing their gene sequence, but also build a library of different promoters, signal peptides, things like this that we can also license to the customers.
Interesting.
Important to be a real part of the workflow. So integrate it into the workflows of major biotech companies. So today, those companies work. They don't know what they are missing because that's how they're working. So it's important for us to convince them that they are missing something and integrate our technology into their workflow. That's why we're also in contact today with also contract research organization doing third-party R&D for those companies, CDMOs who are development and manufacturing companies. In order to push our technology inside, we're in contact with gene synthesis companies who are today, let's say, printing DNA. So it's two development that happened in the past 2 decades is streamlining of gene sequencing. And the second thing is gene synthesis. So today you can write a new DNA and print it in gene synthesis. Important for us is before these companies are printing the gene synthesis, if they can optimize the gene to have a higher protein output, then they will be happy to give this service to their customers. So
I see.
That's an important part of our business. So integrating ourselves into the workflows of the industry and of the biotech companies, and be a standard tool for designing recombinant proteins. So companies will come to us, and from the beginning they will be using our tool to develop the optimized gene sequence and not burning before they coming to us. So we want to be like...
Even solve the problem before it starts, you're saying?
Yes. So what we want to be is top of mind. Okay, when somebody wants to start a project after he developed his protein, we need to be there in their mind just to be used immediately after the stage in order to optimize this gene sequence and increase their chances of success. And at the end, we want to be a key enabler of the fast-growing synthetic biology ecosystem as it scales in the next couple of years to $180 billion. So on the long term, we want to make biology more programmable to give scientists and companies the power to design proteins with confidence from day one.
Hey, wow, this sounds impressive. It was good to hear your story, Steve, and I appreciate you taking the time and sharing it with me and with the listeners, and wishing you and your company a lot of success moving forward.
Thank you very much, and thank you for having me.
That was Steve Grun, the co-founder and CEO of MNDL Bio. We hope you enjoyed this episode. There are many more to come. Do you have a great innovation or startup idea? We'd love to hear from you. You can reach out to us by going to our website, jmbdavis.com. And if you go to jmbdavis.com/startup, you'll see we have a special site specifically made for startups to help startups protect their innovations. Please be in touch with us and find out how we can help you. Thank you for listening, and we look forward to bringing you the next episode.